Hi everyone, today's lesson is about the Central Dogma, and like always, everything is mostly from Campbell's. So this is meant to be about USABO Opens level, but a lot of this stuff could help with AP Biology as well, although it may be a little bit too detailed for that. Alright, so the table of contents is first the basics, second transcription, third post-transcriptional modifications, fourth translation, and 5 mutations. Okay, so first off is the basics. So first we're going to start off with some experiments because like always, people didn't know that much about the nature of biology until they started experimenting. So someone named Archibald Garrod in 1902 suggested that genes were the things that controlled the phenotypes, and they did that through enzymes. And he believed that inherited diseases happened because there were certain enzymes that weren't being made. So he was really the person that brought up an idea that's somewhat similar to what we know about gene expression right now. So there was something else also around that time called the one gene, one enzyme hypothesis, which is essentially that one gene corresponds to one enzyme. And we'll see that this isn't really true, but it makes a decent general model. Okay. So some other researchers, Beadle and Tatum. So they were important. They worked with Neurosporocrasa, which is a bread mold. And what's interesting about this bread mold is that it's haploid. So, it just has one set of chromosomes, and it's made it easy to work with because you don't have to deal with, is it homozygous, recessive, is it heterozygous? It's just going to show that one allele it has. So yeah, and the mold normally can grow on minimal medium, which is just inorganic salts plus glucose plus biotin and agar, which is kind of like this stuff that's got like a bit of a jelly-like substance. It's made from seaweed, and like all of the other nutrients are produced by the mold. So here's the medium. Okay, and then what they did was use X-rays to cause mutations in the molds, and then they studied the mutants for nutritional differences, and then they made it so each of the mutants had like a mutation in one gene that makes an essential nutrient, and all of them could grow on complete medium that gave them everything. So this helped find the synthesis pathways of certain nutrients later. So if you look here, like, there's a mutant that can't grow on minimal medium plus valine, can't grow on minimal medium plus lysine, but it can grow on minimal medium plus arginine, which shows that it's lacking the ability to make arginine. So that's what it was needing, and the wild type would obviously just grow on minimal medium. Okay, so one set of researchers that kind of worked off of what Beadle and Tatum got were Serb and Horowitz, and they looked at the synthesis of arginine. So they essentially made this type of table, and we'll use it. And then, so basically, their predicted synthesis pathway is that from a precursor, we can make ornithine with enzyme A, and then with enzyme B, ornithine turns into citrulline, then citrulline turns into arginine with enzyme C. So this was meant to see, like what mutation do each of these mutants have, like in enzyme A, enzyme B, or enzyme C. So if you look here, the wild type grows on everything: minimal medium plus ornithine, plus citrulline, and plus arginine. So class 1 mutants, they can't grow on minimal medium, but as soon as you give them ornithine, they can grow on everything else behind it. So that tells us that enzyme A is the only one that's not working, because everything after ornithine works perfectly fine. And then same class 2, this time it can't work if you don't give it anything, like if you don't give it anything before citrulline. So that tells us that enzyme B has the issue. And then for class 3 mutants, it only grows if you give it arginine, so the issue is occurring in the last step. Okay, and the next is why one gene, one enzyme is a bad hypothesis. So the first thing is that not all proteins are enzymes. So they changed this to one gene, one protein. And some examples of proteins that aren't enzymes are keratin, which makes up your skin, your hair, and insulin, which helps us absorb sugar. And many proteins are made from polypeptide chains from different genes. So then it became one gene, one polypeptide. And hemoglobin is made from 4 polypeptide chains. So that was kind of, a counter example. And then people were like, but one gene can make different polypeptides. So it's not really true. Okay, so here is the central dogma. It's that DNA through transcription can become RNA, and then from RNA through translation, we can get a protein. So this is what occurs in all organisms. And here's some differences between bacterial and eukaryotic. So for transcription, we call the mRNA produced the primary transcript. And in eukaryotes, we have modifications afterwards, so we just call this the pre-mRNA. And for eukaryotes, this occurs in the nucleus. For prokaryotes, it occurs in the cytoplasm because they don't have a nucleus. And then translation, both eukaryotes and prokaryotes use ribosomes. Okay, so next is codons. So DNA has something called triplet code, where 3 nucleotides codes for 1 amino acid. So if you think about it, 3 is the perfect number, because if we had, say, 2 nucleotides codes for 1 amino acid, then we could have 4 possibilities for the first and 4 for the second. But that means there's only 16 unique combinations, and that's not enough for the 20 amino acids we need to be coding for. However, if we had 3, 4 times 4 times 4 is 64, which is, enough for 20 amino acids. So, how this relates to transcription is that the template strand is the strand being transcribed, and the mRNA is complementary to it. And the bases are the same as if it were DNA replication, except for uracil instead of thymine. And the coding strand would be similar to the mRNA. So this is where we're getting our code. And then so the codons have to do with the triplet code. 3 nucleotides makes 1 codon codes for 1 amino acid. So in translation, codons are read 5' to 3', and 1 codon is an amino acid. And this will make a little bit more sense later once it's put into context. Okay, so continuing on, the way people, researchers matched amino acids and codons were by creating artificial mRNA. So the first thing they created was UUU, so they strung a bunch of uracils together, and then they found out that made a bunch of phenylalanine, so they're like, hey, UUU codes for phenylalanine. And another important concept to know is called a reading frame. So since the nucleotides are grouped into 3s, it's important that you read the correct sets of 3s. So if you look at it with normal words, the red dog ate the bug is not equal to if you shifted the reading frame, so HER space EDD, right? That's not the same message, and it's the same for, mRNA. So UGG, and then just this sequence isn't the same as if you started your groups of 3s with this G or something like that. Okay, and then some special codons to know. The first one is the methionine or the start codon. So it has 2 functions. The first one is to be the start codon. It's AUG, by the way. So it's the codon that starts translation. However, it also functions as a normal amino acid. So, after the start codon, you can have AUG, and that codes for methionine. And stop codons, there are 3, UAG, UAA, and UGA. So, it's important to remember these. And what's important is that they aren't translated twice. So, while there can be multiple methionines or one start codon. Like, so there can be multiple AUGs. One of them is a start codon and multiple methionines. You can't have that with the stop codon, because once you stop, that's it. You're not going beyond that. Okay, next up is transcription. So we'll talk about the molecular components of transcription first. The first thing is RNA polymerase. And if you remember about DNA replication, RNA polymerase basically does what DNA polymerase 1 and 3 and helicase and topoisomerase does. So it does pretty much everything. It doesn't really need much helping it. And, it doesn't need a primer, so that eliminates the need for DNA polymerase 3. And it separates the strands of DNA, and it just goes in, and then the strands close right behind it. So that's what it means when it says helicase and topoisomerase. The functions are covered by RNA polymerase. And it adds nucleotides 5' to 3', like DNA polymerase. And then just some terminology. The promoter is the starting sequence. The terminator is the stopping sequence in bacteria. Downstream is in the direction of transcription, and upstream is in the other direction. So downstream from the promoter would be like the gene, and upstream would be some other things. Okay, so the first thing is initiation. So the promoter has what's called a start point, and this is the nucleotide. And in prokaryotes, RNA polymerase binds directly. However, in eukaryotes, it's more complicated. Transcription factors bind first to RNA polymerase, and then they help RNA polymerase bind, and then transcription starts. So all of this makes a transcription initiation complex, and this happens in the Tata box, which is an area that has a bunch of T's and A's, which is why it's called the Tata box. And once the RNA polymerase is bound in the correct orientation, it unwinds the DNA and begins transcribing. So elongation is, the bulk of the transcribing. So transcription elongates at about 40 nucleotides per second. And only 10 to 20 nucleotides are unwound at the same time, and they close right after, RNA polymerase leaves. And multiple RNA polymerase can synthesize the gene at the same time. All right, so termination. In bacteria, termination is done with a terminator sequence, and in eukaryotes, RNA polymerase instead transcribes a polyadenylation signal sequence, which is AAUAAA in the mRNA, and this is kind of, like the ending point. And then, however, like once it reaches that, it's cut off pretty quickly, and then enzymes catch up to the RNA polymerase, which is still going. So, we get the mRNA cut off, and then we have enzymes stop the RNA polymerase. Okay, next up is post-transcriptional modifications. So it's important to note that these only happen in eukaryotes. So, bacteria and prokaryotes, they don't need to do these things. So at the end of our pre-mRNA, we add a 5' cap at the 5' end. This is a modified guanine. And in the 3' end, we add a poly A tail, which is about 50 to 250 adenines. And the functions are to move the mRNA out of the nucleus to kind of guide it, and then to prevent mRNA from being degraded. So this one, the poly A tail does a lot of that work, because the adenines essentially are a buffer from, the nucleases in your cell that want to just chomp it up, because, there's a chance that it thinks it's foreign RNA. And it also helps the ribosomes attach during translation. Okay, next up is splicing. So splicing is when pieces of mRNA are cut out and the other pieces are put together. So it's important to note what introns and exons are. So eukaryotes have a lot of DNA that doesn't code for proteins, and these are introns. And the exons are, the parts that actually code for amino acids on a finished protein. So a spliceosome does the splicing. So spliceosome consists of protein and some other RNAs. And essentially it just clips this intron out, and then you have a cutout intron, and then you can connect the exons to make, your finished mRNA. which we call a mature RNA. And then something else that happens in this process is alternative RNA splicing. So this happens when you splice out different pieces of RNA to make different mature mRNAs. So if you look here, we have exon 1, 2, 3, and 4, and 5. If we put all of them on, this is what our protein's going to look like. If we leave out 3, it looks like this. If we leave out 4, it looks like this. So just with one gene, you can make multiple proteins. So that's another reason why, one gene, one enzyme isn't a good approximation. And next up is translation. Okay, so we'll talk about the molecular components again, and for this slide, we'll focus on tRNA. So tRNA transfers amino acids onto the growing polypeptide. And what it is actually an RNA strand about 80 nucleotides long that bonds with itself. And so if you see here, this is like the RNA, and it looks like this in like a 2D shape, but in a 3D shape, it actually looks kind of more like that. And it has this area called the anticodon, and here is the place that recognizes this codons in the mRNA, and you read the anticodon 3' to 5', and then they can be in two states basically, charged or uncharged. And then charged is when it has an amino acid attached right here. And the amino acids are matched by amino-acyl tRNA synthetases. And something else to mention is wobble. So wobble is flexible base pairing in the last spot. And you see, like, we have these codons here. So a lot of times it doesn't really matter what base is in the last spot. There's like flexibility for so like this would code for a specific amino acid, but if you change the G to something else, it may still code for the same amino acid. So that's what wobble is. And I forgot to mention this when we were going over codons, but you can use a codon chart or a codon wheel. It's pretty easy to figure out how they work. But basically, U, G, G. Let's look at that codon. So the first letter is U, second is G, and then the third one is G. So UGG. So that makes tryptophan. So that tells us, there is tryptophan attached to this tRNA. So yeah, you need to know how to use a codon chart. They might give you a wheel. It's pretty intuitive, like in using a wheel. Let me go back. There. So first letter is in the center, so U. Second letter, you go to this place, so it's G, and then third letter, you go here, tryptophan. So yeah, it's important to know how to use one of these. You really don't need to memorize any codons except for the stop codons and AUG being methionine. The other ones, like, they'll just give you a chart. Okay, and then a second molecular component is the ribosome. So this is what adds to amino acids to the polypeptide. And it's also made of RNA and protein. And eukaryotic ribosomes are slightly larger than prokaryotic ones. And yeah, it's got two subunits, small ribosomal subunit and large ribosomal subunit. Okay, so initiation is, we form the translation initiation complex. So the small ribosomal subunit binds to the mRNA. And this is actually not a good picture, but I couldn't find a better one. So pretend that this codon is right here. So the small ribosomal subunit binds to mRNA and a tRNA that's carrying methionine. And then the large ribosomal subunits binds, and then the initiation factors, which are proteins, they bring everything together. So yeah, basically we get a small binds to mRNA, and a tRNA that has a methionine, and then the large ribosomal subunit binds. And elongation is, translation elongates, essentially. And there's 3 steps, really. So first step is a codon is recognized, and then Oh yeah, I should mention about the sites on a ribosome. So there is an A site, which is where the amino acid loaded tRNAs come into. You can think of it as A for arrival. I'm pretty sure it's A for amino acyl. So for like the amino acids, and then P for peptide. I think it's like peptidal, and then E for exit. So you can think of it like that. Yeah, so the first step is in the A site, a codon is recognized by a tRNA that's carrying amino acid, and then a peptide bond is formed between the amino acid on the A site and the amino acid on the P site, and then this entire thing will be transferred here. And then translocation is that everything shifts the codon down with a tRNA leaving the E site. So this entire thing is going to go 3 codons over, and then this tRNA will be lost. Okay, and then a termination happens when a stop codon reaches the A site. And stop codons code for a release factor, which is shaped kind of like a tRNA, but it is just a protein. And what happens is the release factors, they hydrolyze the bond between the polypeptide and the tRNA. So it breaks this bond right here. And then it essentially tells everything to scatter. So now we have a polypeptide made, and our ribosome and our mRNAs is just like floating around now. Okay, so after that, we need to finish making our protein. So polypeptides, like they begin folding, as soon as they're being made, basically. And then there are these proteins called chaperonins that actually, prevent them from folding wrong. And there are some post-translational modifications you can make to proteins. Like you can add sugars, lipids, and phosphate groups. You can remove amino acids. You can cut it into different pieces. Which happens with a lot of hormones, and you can also make quaternary structures by combining multiple polypeptides. So, yeah, and then generally something to know is free ribosomes in the cytosol usually make proteins that are, stay in the cytosol, and bound ribosomes usually make proteins that are going to be secreted. Okay, and lastly is mutations. Okay, so the types of mutations, there's a couple, and they sometimes cover parts of each other, so they're not all super clear cut. They overlap a little bit. So a point mutation is a mutation in one nucleotide, and the ones we're looking at right here are all point mutations. So substitutions, so results from substitution is like when you just substitute one for another, one nucleotide for another nucleotide, and it could cause a silent mutation, which doesn't change the phenotype of the organism. So if you think about the wobble position from earlier, the wobble position makes it so multiple different codons would code for the same amino acid. So if it's still the same amino acid, that's a silent mutation. And a missense mutation is when results in a change in amino acids. And a nonsense mutation is when there's also a change in amino acid, but the change is to a stop codon. So it just stops the polypeptide right there. And insertions and deletions. So insertions, when you insert a nucleotide where it's not supposed to be. Deletions when you delete a nucleotide. So this can cause a frame shift mutation. If you remember the discussion about reading frames, if you take away one of the letters and you group them in threes again, it's completely different. So yeah, these are really bad. And here's just kind of looking at a visual representation. So if this is our normal mRNA and our normal protein. So if we had a substitution that substituted an A instead of a G right here, then we end up with a U instead of a C in our mRNA. And although that changes the mRNA, we still are coding for a glycine, so there's no effect on, we're still coding for a glycine, so there's no effect on the amino acid sequence. But if we had a substitution right here, that does change the codon in a way that changes the amino acid. So we get a serine instead. That is missense. Nonsense is if we had a substitution that just resulted in a stop codon, so we just stop. And then insertions and deletions, we can have a frame shift costing, a frame shift mutation causing a nonsense mutation. So if we inserted an extra U here, we'd get a stop codon. So we just stop. And then there's frame shift mutations that cause a lot of missense, where, you know, they shift the reading frame and then everything is different. And then if we have a 3 nucleotide pair deletion or insertion, then we're just inserting or deleting one amino acid. And yeah, now we're going to do some problems. And these are all from past USABO Opens. Okay, this one is from 2017, and just pause the video, take a look at it, and, you know, press play when you're ready to go over it. Okay, so hopefully you've done the problem already, so let's take a look. So if we see here, it says that the influenza genome is copied into mRNA that is like translated into protein. So this is part of the genome. So first, what we're going to do is create that mRNA they're talking about. So if you look at it, this is what it's going to be, right? Just matching it up. This is what the sequence of the mRNA is going to be, and we'll flip it over 5' to 3' for ease, because that's the order we're going to use in translation. And then we can then use the codon table to find out what the protein sequence is going to be. So let's see what ACA gives us. First base is A, second base is C, and third is A. So that gives us threonine. So we know right away it's A, D, or E. I'm not really sure what what this asterisk is, but I'm pretty sure we can narrow it far enough. So next is GUA, valine. So now we know that it has to be D, but let's look at the last one just to be sure. C, G, and U gives us arginine, so we know it must be D. Okay, let's take a look at this one now. This is 2014. So yeah, pause the video and take a look. Okay, so hopefully you've taken a look at it, and what we can see is that C is the correct answer because it's incorrect, because the spliceosome recognizes where to splice because it recognizes that the sequence is an intron, not because of how, the mRNA is. The mRNA is just a strand, and, the spliceosome just scans it for, sites that tell it, hey, this is an intron, so it can take the intron out. Okay, and take a look at this question. Okay, so the answer here is D. The entire chain of amino acids exits the ribosome before it begins folding into its 3D shape. That's not true. We actually have to have proteins to kind of prevent it from folding right away, because it wants to fold right away. And that kind of ties into answer choice C. So just know that it wants to fold right away, and we actually kind of have to keep it from folding right away. So D is the right answer. And C was that the 3D shape of a protein is largely determined largely by its amino acid sequence. That's true because, whether the amino acids are hydrophobic, whether they're polar, if they're acidic or charged or anything, that determines how they're going to interact with each other. So a lot of what the protein's shaped like is based on how close the amino acids want to get with each other. And if we look at A, all protein synthesis is initiated in the cytoplasm. That's true. B, many copies of a protein can be synthesized simultaneously from the same RNA. That's true as well. I did mention this, but you end up with something called a polyribosome, where you have an mRNA with a lot of ribosomes on it that are synthesizing proteins. And then E, the first encoded amino acid in every protein is methionine. That's true because that's the start codon. And yeah, thank you so much. I hope this was helpful. Please leave a like if it was, and please subscribe for similar content. See you next time.